Hello, my name is Nash Turley, and I got a short presentation here about some research we've done looking at the effects of past agricultural land use and more recent restoration activities on soil, fungi, and bacteria. If anyone actually watches this, let me know on Twitter. I'm at Nash Turley, or send me an email. So first, I'd like to acknowledge my co-authors here. It's Lars and Sarah and Lucas, and then some funding from the U.S. Forest Service. Now, about 40% of the world's land is used for agriculture in some way or another. So we have this massive footprint that's impacting biodiversity. But then those impacts of biodiversity are not just active agriculture. There's also lots of areas that were in agriculture that were abandoned in some way. So there's an estimated area of abandoned agriculture about 3 times the size of Texas. And there can be really long legacies of agriculture in those regions of changes in the soil and different plant communities, all sorts of long-lasting effects that can last centuries, millennia even. So with that in mind, the main 3 things we were interested in looking at were, do we see these agricultural legacies influencing soil microbes, their diversity and composition? Does more recent restoration also impact soil microbes? And do those restoration efforts overcome the legacies of land use history on microbial biodiversity? So we did this work in longleaf pine savannas, which is an ecosystem that stretches across the U.S., the southeastern U.S. It's an incredibly endangered ecosystem with at most about 3% left. It's also incredibly diverse with up to 900 endemic plant species and is considered a world biodiversity hotspot. So it's been really heavily impacted by agriculture, but there are some areas that were in agriculture that have been abandoned, which are opportunities to restore, to promote native biodiversity. We worked at the Savannah River Site, which is in South Carolina. It's a really big area, about 80,000 hectares. And you can see it on this map with the surrounding is lots of agriculture, but inside it's very forested and more natural. So it's a very unique place to work because the U.S. Department of Energy developed this site to create nuclear weapons in the 50s, so to enrich uranium and do lots of other secret stuff. So to do that, they had to kick everyone off. So this was an area that was full of people and towns and agriculture, and then all of a sudden in the 1950s, it was all abandoned and then was managed by the Forest Service. So it was this natural experiment of sorts to look at what happens when agriculture is abandoned. So here's some maps of what it looks like. So in 1951, these all these white areas were agriculture, and little red dots here are patches of remnant longleaf pine savanna. So areas that never had agriculture. And then fast forward to present day, all the areas that were agriculture are reforested, mostly with pine plantation, but these little patches of remnants still exist. And many of these remnants were right adjacent to where there was agriculture. So this created a great opportunity to study the differences between remnant areas and areas that were in agriculture that were abandoned. So across Savannah River Site, we selected 27 locations where there was a patch of remnant here, so that there was never in agriculture, that was immediately adjacent to a place that was in agriculture, but then was abandoned now over 60 years ago. Anyway, so there's 27 replicates where we have this natural experiment of a remnant longleaf pine savanna next to a post-agricultural area. So here's some looks at what that looks like. The remnants are now mostly broadleaf forest, and this is because of decades of fire suppression. So in the absence of fire, you get encroachment of all these broadleaf. So we have lots of savanna plants growing in these areas, but it doesn't look a whole lot like a savanna because of fire suppression. And then the post-ag areas were planted as pine plantation and also have this lots of fire suppression. And the leaf litter can be very different. We have all these broadleaves in the remnants and thick pine straw in the post-ag areas. So on top of that, we wanted to then look at restoration. So at each of these sites, half of the plots were then restored, and we did that by removing the trees. So it's a restoration thinning to restore the open savanna-like structure that was natural to this area and that is then maintained by fire. So then we have 4 different treatments. We have remnants control, which are broadleaf forests, post-ag control, which are loblolly pine plantations, and then these thinned remnants and thin post-ag areas. So here's the full experiment. And so what we did across these 4 treatment types is I went out and collected soil from each one. So in each 1 hectare plot, these yellow spots are where I collected soil and then clumped them all together. So I had 1 soil sample per 1 hectare plot with 4 different treatments at each site. So that was over 120 individual plots total. So here I am out collecting soil samples. Here's all the samples, and then we packaged them up in tubes. And then I won't talk too much about this, but we went and did some metagenomics magic to extract DNA and allowed us to estimate the communities of bacteria and fungi that were in those soils. We also took a lot of environmental measures that I won't talk about today, but that's part of the story as well. So revisiting these questions, do those agricultural legacies influence biodiversity? Do those restoration treatments or restoration thinning also impact the soil microbes? And does that restoration impact the legacy of the land use effects? So we'll have a few figures like this. So this is looking at the microbe biodiversity. So this is a measure of Simpson's diversity. And then we have our 4 different treatments. There's remnant, post-ag, control thinned. The colors are all consistent throughout all these. The blues are the remnants and the reds are the post-ag. So the first result is that the land use effect had effects on both bacteria and fungi, but they were opposite. So in bacteria, this history of agriculture increased bacterial diversity by 54%. But in fungi, the legacy of agriculture decreased diversity by 18%. So basically, past human land use is increasing bacterial diversity and decreasing fungal diversity. Now let's look at restoration. So restoration, going from the solid points to these open points, increased bacterial diversity by 14% and increased fungal diversity by 60%. And then looking at all the plots together, we can see that the restoration had similar effects in both remnant and post-ag. So in both land use histories, it was increasing diversity. So let's look at composition. So this is an ordination. And so basically how this works is we had the same color scheme as before. The reds are post-ag and the blue are remnants. And the way this works is that if 2 points are closer to each other, that means they have very similar compositions of species, quote species, and they're really operational taxonomic units. But we can think of them as species. So points that are really close together have very similar collections of species, and points that are further apart have different species. So we can see what we have is 4 different communities. The land use is affecting composition and restoration is affecting composition, resulting in 4 different communities. And so both land use and restoration are having effects, and restoration does not overcome the history of these agricultural legacies. And we see similar results in bacteria, although the results are strongest in fungi. So to just revisit and answer those questions, we found that absolutely agricultural legacies have big effects on soil biodiversity. They're tending to increase bacterial diversity and decrease fungal diversity. The reasons for that are not particularly clear, but that's probably, from our evidence, has most to do with below ground aspects like soil nutrients and stuff like that. And restoration does have really big effects on microbe diversity as well. And our evidence suggests this is primarily due to above ground things like plant diversity and leaf litter, stuff like that. And then finally, the restoration is not overcoming this legacy of land use. So it shows that, like in many other systems, it's going to be pretty difficult, even with active restoration, to try to overcome this legacy of past land use. So if you're interested in more about this, this is in a paper in Journal of Applied Ecology. And get in touch with me on Twitter or email. Let me know if you have any questions. Thank you very much.